Hey, good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps Research. Let me be the first to welcome you here to our webinar featuring Professor Andrew Ward. I would like to say a few things before we get going. Some of you might be new to Scripps, and I'd like to just say a few words about the Institute and what's going on here and our efforts in particular in the area of mitigating coronavirus. So we have two campuses, one in La Jolla. I'm here in my office in La Jolla, and we have a sister campus in Jupiter, Florida. There are 200 faculty between the two campuses that are distributed across 5 departments. We have chemistry, immunology, structural biology, neuroscience, and molecular medicine. So Andrew is a member of the structural biology department. We also have a remarkable and accredited PhD program. We have 200 students. We're especially proud of our national ranking in chemistry and biology. And it's particularly gratifying. Andrew is actually an alumnus of our PhD program. So we are populating top institutions around the world with our student alumni. One of the things that makes Scripps very special and well poised to combat the coronavirus pandemic are all of the faculty members with decades of experience on research in virology and viral therapeutics. In particular, we have a lot of expertise in HIV and flu, and all of the approaches that have been put in place here are now ready to be poised to take some action against the coronavirus. Little good news on the drug discovery front. So this past quarter, Zeposia, also known as ozanimod, was approved by the FDA to treat relapsing forms of multiple sclerosis. And we also have another indication for ozanimod, which is multiple sclerosis. And the phase 3 clinical trials results were just announced, and that is actually affects even more people than the MS market. So this is really a terrific impact that the drug discovery programs here at Scripps are having on human health. But for today, we're going to focus on the coronavirus, and this is a part of a global health initiative combating infectious disease in many different aspects around the world. It's a collaboration between our main basic research engine at Scripps Research. The drug discovery arm is Caliber, and we have a translational research arm, which is SRTI. The Global Health Initiative has 3 pillars. We are looking at ways to detect infectious disease, to prevent it, and then to treat it. So we've had lectures in the front row series addressing all of these aspects in our battle against coronavirus. And I'll just quickly review. We've had Christian Anderson, who's an epidemiologist. He uses DNA sequencing to track viral outbreaks. We've had Mike Farzan, who is looking at antibodies that bind to the spike protein and trying to understand how the virus is evading our immune system. We have Arnab Chatterjee, who's part of Caliber, and he is doing drug screening to try to rapidly identify therapeutics that can be used. And this is taken from a collection of all the known drugs in the world. And it's called the Reframe Library. And we try, these are all approved for use in humans. So if you find something that hits coronavirus, it can be rapidly deployed as an antiviral therapeutic. We heard from Dennis Burton. He's collecting antibodies from survivors of coronavirus and trying to understand how to use those to both treat patients and to develop new vaccines. We heard from Eric Topol and Dr. Jennifer Radin. They were using personal digital medicine to actually identify people who might be experiencing coronavirus from their personal fitness devices. And then brings me to Andrew Ward, the lecturer for today. So Andrew's been here for 20 years. He started as a graduate student in our program in 2001. He got his PhD here and continued his postdoctoral work. And on the basis of all of that work, he was appointed to the faculty as an assistant professor in 2010, and he rapidly rose through the ranks to full professor in 2017. Andrew is a structural biologist. He is using electron microscopy and X-ray crystallography to study the structure of membrane proteins and, most recently, antibodies that are binding to the surface proteins on infectious disease agents. So he's been looking at HIV, malaria, Ebola, influenza, and now coronavirus. So he clearly isn't afraid to tread in the face of the most daunting infectious diseases. And the work that he does helps us understand how our immune system is recognizing these pathogens and how we might improve antibodies and therapeutics to combat these infectious diseases. So it's my pleasure to introduce our alum and our faculty member, Andrew Ward.
Hello, everyone. Thank you, Jamie, for the introduction. And welcome, everyone, to this front row lecture. I'd like to extend a special welcome to friends, family, neighbors, and other members of the community that we don't get to engage with on a regular basis. And this is a really great opportunity to share what we do and provide some context for what happens in our daily grind, in our daily job. And so today, I'm going to take some time and share really most of the historical work we've done for different viruses and how that's fed into what we're doing now with coronaviruses in vaccine development, and also just offer a more global picture of all the efforts that are quite different in vaccine development, but all converging on some common themes in this really unprecedented experiment on a global scale, right? This is a global airborne pandemic that we all need to take seriously and combat. And it really is a collection of scientists, both within the Institute and outside the Institute that are driving this. Just a couple, personal notes. Again, family, I'm really happy that parents and family can join in a way that they haven't before. Just to clear up any clutter on the chat room, my Justin Bieber hairstyle is a direct casualty of COVID-19. I haven't got a haircut in like 4 months. So hopefully that answers a lot of the questions that I might have. And to my lab, go easy on me on the Slack channel. I'm sure you'll be roasting me the whole time. So with that, I'll dive right in. So this is my lab, or almost all of my lab, back in the days when we could actually get together and take a picture like this. And it's really the young scientists that are driving the various aspects of our research. So as Jamie mentioned, we do a huge diversity of projects, and as such, we recruit diverse talent and background from all over America and all over the world. And each one of them is using a different, angle and aspect to work on different projects, such as vaccine design. And importantly, this group, has weathered the storm during partial and full shutdowns, and some members have really stepped up and continued our effort during these hard times. And then I'd like to also acknowledge, a tremendous amount of financial support from both Scripps Research, as well as the Bill and Melinda Gates Foundation and the National Institute of Health, who fund a lot of the work we do. So the government funding, as well as the philanthropic funding, actually is funding things that may not have a direct application now, but as you'll see and I'll share, have direct applications down the road. And this sort of vision to invest in the future is really important. And again, my role as the mentor investigator is really to bring in this talent, put them in a great environment, polish off their graduate or postdoctoral training, and then set them out into the world to do great things and to be future leaders in science. So it's a really pleasure to be here. And again, as Jamie said, as alumni of Scripps, it's quite a source of pride for me that I'm now part of the system. So that was our group in friendlier times. This is what our group looks like now. It's more of a collection of individuals. And so I'd like to just acknowledge this is John Torres, who is our wonderful photographer in the lab, and he's helping document what it's like to do science in this day and age. And so despite what you see on the beaches or in some states opening up restaurants and bars, we take this very seriously. So we practice social distancing, we wear masks, we have a lot of precautions in place, including color-coded gloves and other fashion statements, which help us to prevent spread internally. So we want to be really careful because this is an airborne infectious agent, that we can continue our important research and we don't negatively impact our neighbors. And so it's really, again, a testament to the dedication of the folks in the lab that show up every day, that get temperature checks, that wear masks, et cetera. And also taking on new roles, from John being the new COVID coordinator in our lab to Hannah, taking on a lot of additional roles and helping our facility run. So despite all these masks, what's really nice, and as you can appreciate from this picture here, this is Alex, a postdoc in the lab, and you can see that even under that mask, he's smiling. So we're happy and to be back, and everyone is going sort of full effort, not only on coronavirus, but back to a lot of the other related projects that are going on. So as Jamie mentioned, we primarily use cryo-electron microscopy, and that's just a sort of long way to say, this fancy electron microscopes or microscopes that allow us to image very small things to atomic level details. So instead of light, we use electrons, and their short wavelength allows us to reveal the underlying atomic details. Scripps has invested in this technology historically, as well as looking into the future. And as such, we have one of the best facilities in the world. And so it's really this wonderful backdrop to do science, and we can pivot and do research from neuroscience to vaccinology using this wonderful equipment. And it's also supported by pretty fantastic personnel such as Bill Anderson, our facility manager with more than 35 years of experience that really wrangles an enormous amount of technology and keeps it running, and people like Hannah who load all the samples onto our microscopes and make sure everyone's project is moving forward. So this is our control room, and it's a really great place. It was well designed to give tours. We're not giving tours anymore at the moment, but this is where we remotely control these microscopes. And then back behind this piece of glass is our centerpiece, our $5 million Titan Krios microscope, which is larger than life. So just to give you some perspective, when my daughter was 2 years old, I tried to load her into the microscope. Didn't really work out so well, but you can appreciate just how large these pieces of technology are that are really doing cutting edge science. Behind the scenes, we've invested a lot in really advanced software and in high performance computing. And that's because we collect terabytes of data every single day. This is a data heavy technology. And so at the Institute, our high performance computing manager, JC Dukem, and a staff scientist in my lab, Charlie Bowman, have really designed and built a wonderful architecture that allow us to move terabytes and petabytes really of information around in order to do the science that we need to do. And really, buying and using enterprise grade GPUs and other computing resources. And then importantly, for fidelity and storage and backup is a real big problem. And so, we take for granted, when I did my PhD, I actually took images on the microscope and had to print out the films. A lot has changed. And the example that Charlie likes to use is if we're able to collect on these microscopes and these really fancy cameras up to 1,500 frames per second, that's about, if you were to take that data and print it out on micrographs, you'd get about 1.25 feet per second, or in about 6.5 hours, that's the height of Mount Everest. Charlie packs that down nicely into 7 inches of hard drive. But it really is a remarkable ability for us to generate that data and then store and analyze that data. And we're learning, especially with new approaches in machine learning or artificial intelligence, how to extract even more information from that data and get more bang for our buck with what we're doing. And the end product is really something that looks like this. So this is just a gallery of images that I put together to highlight some of the areas that my lab is working in, from HIV, Ebola, malaria, influenza, and coronavirus. And there's a certain aesthetic appeal that I hope you can appreciate from all these images. And more importantly, what is not as obvious from the images is the atomic and molecular level detail that reveals, for instance, how antibodies neutralize a virus and how we can use that information to either design better antibodies or, more importantly, vaccines that can elicit those antibodies in people. So the great part about being a structural biologist is there's a certain aesthetic and certain artistic element that I think draws in a lot of people, and also becomes intuitive after you start looking at these things for a while. That's a little bit more intuitive than for, say, looking at sequences of proteins. So we, as Jamie mentioned, have been interested early on in my lab at really using the structure pipeline that we have to understand how antibodies, primarily from infection, neutralize or somehow otherwise can attack a virus or bacteria for that matter. And really those lessons start from collaborations with clinicians in endemic areas or local areas. Here's just a nice graphical abstract showing, here's an example when we were working on Ebola outbreaks in Uganda. We had local clinicians that were isolating B cells. These B cells are the cells that make antibodies. These cells came back to America to a collaborating lab at Vanderbilt. They made a bunch of these antibodies from B cells. They then came into our lab and we started solving structures. And what we can do is we can start to dissect out some of the differences in antibodies and start to understand what makes a good antibody or a bad antibody. What makes something that only neutralizes this one single Bundibugyo strain of a filovirus, Ebola virus, or an antibody that has a different phenotype that neutralizes multiple filoviruses at the same time. And so this is a really well-trodden established pipeline. And we were one of the first labs to really start using the breakthroughs in electron microscopy to accelerate this process. And we're using it now more than ever. So as a really a pertinent example of the impact, I use this example from a few years ago. So Francis Collins, who's the director of the NIH, in his blog in 2014, wrote a little bit about the structural work that was going on in my lab. That was also a collaboration with MapBio in the Sapphire lab. And this is an image of ZMAP. So each of the colors is a different antibody. This was the triple antibody therapy that was used in the Ebola outbreak for compassionate care early on. And it parallels some of the efforts that are going on for coronavirus now to develop antibody cocktails that can be used to treat coronavirus infection. And additionally, in his testimony to Congress to justify the $31 billion budget of the National Institute of Health, he stated, or he referenced our work as a compelling current example of how basic science is revolutionizing the treatment of emerging diseases like Ebola. So that really shines a light on how impactful this type of research can be, and so we've been very fortunate to receive this level of attention, and we've been using it to look at a lot of different viruses. So this is just another aesthetically pleasing gallery of structures. So this is from Daniel's thesis, his PhD thesis. He was a student at Scripps. And it shows you just a sampling of some of the different antibodies that we imaged or solved the structure of to combat different filoviruses, Ebola viruses, et cetera. And you can see there's a huge diversity in the way in which they approach the spike protein on the surface of Ebola, and therefore have different mechanisms for neutralization or function that makes them interesting or not interesting targets for therapeutics or for vaccines. So the reason that we use this information and why it's important is that we are sort of entered a new age in vaccinology. So historically, examples like polio vaccine are really a pretty simple solution despite an enormously complex biology that still isn't fully understood. So in a polio vaccine, the infectious particle is grown up. It's then treated with some chemicals or irradiated to make it non-infectious anymore. So it has sort of the skeleton or shell of the virus without being infectious or dangerous. And it has a lot of other things in it that are immune stimulants or agents for inactivation, et cetera. When this is injected, the global population responds very well. And in fact, we have a really great vaccine. And this was done using empirical methods, and it works really well. In the sort of modern world, against a lot of the pathogens that we look at in the lab, you can't just do that. So here's an example. Here's HIV. If you inactivate HIV and inject it in people, they don't develop a robust immune response. There's many different reasons for that. But what we focus on is that there are pieces of the virus. So here's just another high resolution image where you see there are these spike proteins, these red proteins on the surface. We know that these are the targets of neutralizing antibodies. So again, by looking at these at very high magnification, high level of detail, we solve atomic level structures. And from those atomic level structures, we can design vaccines that either are the whole piece or some engineered piece. And really, we're trying to extrapolate these atomic details to focus immune responses on really the key sites and targets that we can really synergize the immune system and focus the immune system to get the response that we want. And so, just another sort of example and sort of blowout, this is what most people think of when they think of vaccines. It's a syringe. You get something that ideally leads to robust and durable immunity. But inside that vaccine can be something like an inactivated virion, similar to polio. You can have a protein subunit vaccine, and that's just one piece of the virus, the piece of the virus that's really the key site of vulnerability. You can also deliver the genetic material, mRNA or DNA, that will enable your body to make those pieces of the virus and therefore generate an immune response against that. And then there are vectored ways to deliver genetic material, again, to make these proteins in your body. And I'll give you a few examples later on of how all of these different platforms are being developed. And really, we're moving beyond the inactivated vaccine, which is the basis of a lot of the vaccines that you received in childhood, more towards these type of approaches up here. And structure, molecular biology, bioinformatics play a huge role in how these are designed and deployed. So COVID-19 is a particularly interesting experiment at a global scale. There's well over 100 vaccine concepts now. And sort of the most important point to make is that they are all focused on the spike protein. So that, again, this is analogous to the HIV spike or the Ebola spike, which I'll discuss in a little bit more detail. And you can see there are different phases of clinical development. Several have been into humans, and now it is really a matter of scaling up and figuring out which ones are working, which ones aren't, and maybe tuning them up a little bit and re-engineering them. So it's an interesting time because in no other vaccine space is there such a vibrant activity with different technologies being used. So again, sort of a historical backdrop. My lab became interested in coronaviruses about 2015, which coincided with a few different things. So in 2015, Bill Gates gave a great TED Talk about how, the thing he's most concerned about, and he was quite outspoken, is still quite outspoken about the role that a potentially airborne infectious virus could have in driving a global pandemic, which has really come to fruition here. The Gates Foundation supported the Coalition for Epidemic Preparedness, and that was a huge investment, and that investment is now funding a lot of the vaccine development, like companies like Novavax. So it was sort of forward thinking, and the reason that we had a sort of basis, as well as Gates and others, is that coronaviruses have a lot of parallels to influenza, and they can circulate in animal reservoirs. For coronavirus, it's bats, pangolins, camels, etc. Every so often, there's some evolution that happens in those animals that causes an outbreak in humans, right? And so we became interested in particular postdoc, Rob Kirchhofer, came to the lab interested in coronaviruses, and he's actually since gone on and started his own lab at University of Wisconsin, still working on coronaviruses. And he came to the lab and said, well, what can we do? And we said, well, maybe we can learn about these viruses by studying the structure of the spike protein. And there's the seasonal versions. So most people don't know that they get infected seasonally with these common cold causing viruses, which are fairly mild and aren't so pathogenic. And then most of you have heard of SARS and MERS, these viruses that are much more pathogenic, but didn't really have even though there's a big threat, they didn't pan out. SARS-CoV-2, unfortunately, had a few adaptations that's made it, really into the pandemic that we're seeing today. So spike is the major surface protein, as I've mentioned a few times. And so what's important about the spike is it has both the ability to recognize the host cells to infect, as well as the machinery to get that virus inside the cell, deliver its genome, and make new copies of itself and go on infecting other cells. And so again, a lot of historical studies showed that this was a functionally important protein. Here's just showing you a schematic of what that looks like. And so from these cartoons and from the schematics, we can understand something about the structure and the domains and other pieces of the virus. But explicit atomic level structure description really provides the details that we need to investigate deeper. And just a shout out to Haley, who's a first year graduate student in the lab, who created a bunch of these beautiful graphics that were featured on our website as sort of an educational outreach. So spike is the target. It's been shown and is being actively shown in a lot of different studies now that the spike protein is the target. And this is the basis of a lot of the vaccine design that happened at great speed. So that these antibodies can do a couple different things. They can directly compete with or bind where the ACE2 receptor would bind. And so that the virus then becomes coated with these antibodies and can't get into the cells. There's other mechanisms by which they disable this sort of triggered, or this hair triggered mousetrap, which is the fusion machinery, this little molecular machine that allows the virus to fuse with the host and get into the host. So as I mentioned, Rob Kirchhofer as a postdoc was looking for interesting projects, and then our collaborators, Jason McClellan and Barney Graham, had common interest in coronaviruses. We were thinking ahead of that these might be a problem in the future, so we wanted to start understanding them. So we got together and used our different backgrounds and technologies. We were using cryo-EM to really drive this. And this is what the first structure looks like. So we solved the structure of a human HKU1 coronavirus. This causes seasonal colds. It's a trimer. So there's 3 copies of it here, and it's standing up on its side here. And it can be further decomposed or broken down into 2 domains. So S1, and this is where the receptor binding domain is. So this recognizes the cells that it wants to get into and infect. And then here's the fusion machine, this sort of mousetrap spring-loaded mechanism that once it finds the right cell undergoes a very large conformational or structural transition in order to create the energy needed to get into the cell. And so you may ask, why were you looking at the seasonal coronavirus instead of the more pathogenic ones? And the answer is because it worked. So oftentimes in biology, you have a target or a set of targets to look at, and the ones you want to look at don't work. But it's important, as I'll describe in the next slide, why we looked at this one. So once we had the structure and we understood something about this very unstable hair-triggered fusion machine, we could start making mutations and do analysis at the bioinformatic and sequence level in order to make mutations that would then prevent that high energy structural transition from happening in other spike proteins. So we made tens of mutations. We screened them using different technologies. And you can see we did try to make MERS and SARS early on. And here's just a gel filtration or a way in which we purify the protein and measure how much we're getting. You can see there's almost no protein. But when we add these two proline mutations that prevent this structural transition, all of a sudden we get a lot more yield in the protein. We then also change the phenotype of the structure that is underlying. So here you can see if we don't make mutations in MERS or SARS, we get a mixture of these pre-fusion and post-fusion conformations. The red ones are bad and off target. We don't want those. The blue is what we want. So that making these mutations really enabled us to then study and characterize these. And we also showed that these mutations work for lots of other coronaviruses. And so we could predict then that these could be used in order to make pre-fusion coronavirus spikes, which would be useful for structural and other studies, but also potentially for vaccines. This was eventually patented, and actually these mutations are in several of the vaccine platforms. So this prior knowledge allowed a lot of people in the vaccine development space to rapidly introduce those 2P mutations and then accelerate their vaccine platforms. And so we use those 2P mutations in order to solve structures of MERS and SARS and CoV-2. And each one of these structures is an opportunity to learn and inform vaccine design, maybe make additional stabilizing mutations or do other things. So the knowledge that we've created, with some forward-looking thought is now being implemented, which is really cool to see and really shows you how powerful basic research can be. And so this is just a summary of that really the prior knowledge allowed the rapid development in companies and other institutes to be prepared to rapidly make this spike protein, and then a lot of other developments in vaccine platforms for other viruses were then able to be converted into the SARS-2. So once the genome was available, people could do the bioinformatic analysis, put everything together, and that's how you're seeing such rapid development of vaccines. And here's just a timeline just showing that, in January 11th, when Chinese scientists made the full genome available, it was very soon after that, a month later, that the first structures came out. Moderna had already entered phase 1 by March 16th. And it's again because of this prior knowledge. And so it's really unprecedented, the pace at which we're going, and it's really necessary, of course, in this case. So, we're continuing to do a lot of structural biology. And so what we'd like to know and understand is how the differences, the small differences between SARS and SARS-2 spike are driving some of the pathogenicity and other, of the more deleterious effects of this virus. And so, we can locate now in 3 dimensions where this multi-basic cleavage site. So this is a sequence that was inserted into the genome that enables the spike to be processed better, but it also has a huge impact on getting into human cells. And we also know, where the receptor binding site is and how that's accessible for antibodies. There's also another mutation that's been actively studied where an aspartic acid goes to a glycine, and that's really been, it seems like that makes the virus spread better, and so you're seeing that sequence come up more and more in the epidemiology studies. And so again, we can provide the structural and molecular understanding of how this deletion of 7 atoms out of over 25,000 atoms may have an impact on the infectability of the virus. And Sandhya in the lab is really leading the charge on this and doing a great job of wrangling an enormous amount of structural work that's ongoing. So now I'd like to just kind of blow it out a little bit, show you a few different of the vaccine strategies, and kind of give a quick overview. So I've talked a lot about how the spike is the focus, the focal point. We know a lot about the structure now, and in particular the receptor binding domain. At the time, most people were pursuing just the entire spike as a target for vaccine development, and again across very different platforms. And they all have different strengths and weaknesses, some of which I'll allude to, but they're all done remarkably quickly. And there's a lot of different estimates on timeframes. I think that's still a huge unanswered question of scalability of some of these vaccines, particularly as efficacy and other trials are only starting to be done right now. So mRNA and DNA, they're fast but full of unknowns, right? So it's very easy for Moderna to take all the historical knowledge and implement a vaccine and to synthesize the nucleic acid or genetic material required to make the spike protein. Some of the sort of downsides are that there's no currently licensed vaccines in this space. When you inject this and you ask the cells of the body to make the spike protein, you don't have any quality control, so it could be making lots of aberrant or other versions of the spike protein, which may distract the immune response. Adding those stabilizing mutations that I mentioned probably ameliorate some of those effects, but again, it's still unknown right now. A vectored vaccine is nice because what you're doing is you're coding genetic material and you're delivering the genetic material in a vector or virus that's not really infectious. So adenovirus is a popular platform to get into the human body. You might have pre-existing immunity to the actual vectored virus, and that could create some problems. And then again, by making the spike protein in vivo, you have less control over the quality. Recombinant protein vaccines, and that's where we're most active, are really afford you a high level of quality control. So it's an additional manufacturing step, a pretty huge and costly manufacturing step. But what it means is that the material that goes into the body is just the piece of the virus, the spike that we're interested in, and it's highly controlled for different properties. There's a few more recent FDA-approved precedents: influenza, hepatitis B, human papillomavirus, which is a huge success. The one sort of key factor in these is that they require adjuvants. Adjuvants are co-immunostimulatory molecules, and this is a huge space that's under active investigation right now. And then inactivated or live vaccines are also being pursued, and they're going to be a good sort of control for a lot of the other platforms that are going on, right? So as I mentioned, there's many FDA-approved and effective vaccines in this space. There are, of course, safety concerns anytime you're working with a live attenuated or some other killed virus. And then more importantly, when you chemically or radiologically inactivate the virus, you can damage the spike protein, so you're not putting in the spike you want. So there's quality control issues. And then the upside is with the whole virus going in, you do stimulate multiple arms of immune response, and it's most similar to infection. And infection really gives you a really strong level of immunity typically. So here's just one sort of quick advertisement example of, again, what we're doing. This is with our collaborators in Amsterdam who were working on HIV with us. They pivoted just like we did to work on coronavirus in pretty rapid order. They took 3 patients, 2 really good responders, 1 less good responder, isolated monoclonal antibodies. Each one of these is a monoclonal antibody. Most of the antibodies that they isolated did nothing. They weren't neutralizing and sort of were off target. A subset were. Those came to us for structure determination. And again, you can see here's the sort of range of antibodies that we saw when John and Sandhya imaged them. And you can see that most of them actually, the neutralizing antibodies bind to and really prevent the ACE2 receptor from engaging the spike. And so, this new knowledge is really shining a light, as is many other studies like from Dennis Burton here at Scripps, that the receptor binding domain, not the entire spike, is really the key immunogenic target. There's a nice convergence, at least in convalescent and recovered patients, as well as some other studies. Importantly, this one antibody, COVA-215, which is very potent, binds to the receptor binding domain no matter the conformation. So that receptor binding domain can go up and down and kind of act as an immune evasion mechanism. But this antibody binds both conformations, saturates the spike, and really does not allow the virus to get into cells. The good news is it's relatively common, and more studies of patients are uncovering these type of antibodies. And there's 3 science papers this last week that showed this. And then, I think the working hypothesis is that if we can elicit these type of antibodies, they're likely to be protective in patients. So again, we're learning as we're going. And the spike protein is now really the focal point is just this receptor binding domain, even though all the vaccines that were developed early were focused on the entire spike. So there's new programs just focusing on the receptor binding domain. And that's important, again, because if we don't have, if the virus is not allowed to bind ACE2, it can't get in and is sterilizing immunity. It's the type of immunity you want, because if it gets into the cell and then you have to treat or attack, there can be additional complications. So what we do know, again, through our mapping studies of seasonal and other coronaviruses, is that antibody responses are quite diverse, so quite similar to what we've seen in all the other viruses that we've looked at. And so the outstanding questions are, what are the most effective antibodies? I think, as I just said, the RBD antibodies are the ones we want. But that creates a challenge in how we elicit those either specifically or preferentially. While all the vaccines have a version of the spike and therefore some or a version of the receptor binding domain, or the RBD, we don't know at what level we'll get RBD antibodies versus maybe other off-target antibodies. I think it's important to consider, different populations and demographics, aged population, young population, will they generate the same type of antibodies, and how durable will be the response? We're very poised and actively seeking out collaborations to start now mapping the antibody responses to the different vaccine platforms, so we can understand which ones might be doing better or worse. And really, it's, most of the data that's come out from the different vaccines in humans, is encouraging, but not hugely impressive or overwhelming. And we really need more data in order to understand that. And I think we're on a timeline, that is in the years of maybe even having to refine the vaccines a little bit further. And so that's a sort of a rapid run through how we've participated in this process, how some of our early research has driven what's going on right now, and what we're doing actively in the lab to map antibody responses and inform and improve the vaccines that are out there. And as I noted at the beginning, basic research is really driving most of this vaccine development. So industry and pharma is picking up the lessons learned from HIV and coronaviruses and other places, and implementing their pipelines at what is an unprecedented pace. But still, is a longer term vision. I don't think any of us think that even though you might be able to make the doses and do some trials, there's a lot to be done in terms of efficacy trials and then dissecting which ones work in which populations, so on and so forth. But the really, again, the vision and why we do science is that the lessons learned now from this outbreak and pandemic will definitely be used to combat future pandemics. And we'll know, should we start within, an mRNA-based platform? Should we go directly to protein subunit vaccines? And that's really how science works, is accreting lessons from the past, refining the techniques. And I think as I've shown, and really what's been the focus of the lab, is taking advantage of technological and computational developments to be able to do this even faster in the future. So with that, I will turn it over to Jamie and questions. And I really appreciate everyone joining. And I hope you have some appreciation now of what we do and the sort of art of structural biology and how it plays a role in vaccine development. Thank you.
All right, thanks a lot, Andrew. Terrific lecture. So I think there's a lot of interest in general, and you did speak to this a little bit, but I think it's worth maybe discussing, the timeline for making a vaccine. And I think as you described, there's first you have to demonstrate safety and then efficacy, and then you have to scale up to hundreds of millions, if not billions, of doses. And it's really amazing the pace that we're seeing things moving, and yet it's not fast enough. So what's your best guess? When are we going to actually have a vaccine?
So my best guess is that we are going to have a handful of safe but questionably efficacious vaccines within the next 12 to 18 months. And that will be, after these larger scale, tens of thousands of people clinical trials occur. At that point, it's going to be difficult to probably pick a winner, right? Some are going to elicit neutralizing antibodies, or many are going to elicit some level of RBD, directed neutralizing antibodies. We don't know about the durability, right? And that's hard to say. Durability means time goes by and then you study how protective it is. So time is an element. So I think we won't know anything except for a year after that. So I think, again, we're going to have a handful of things that are ready to go into humans, that are likely safe, that have some, effect, and then it will come down to manufacturing. So mRNA, DNA vaccines, there's no licensed vaccines. One of the reasons for that is, even though it's easy to manufacture nucleic acid vaccines, it's very hard to package them in the sort of delivery vehicles that they need, and they've never been deployed at that scale. There's also stability issues and other things. So you're really looking at whatever we do, it's as fast as humanly possible, technologically possible at this point. But I think, it's a matter of a couple years before we are much more confident in choosing one or multiple candidates as the vaccine.
Right. It's remarkable. Every virus is different. There is yet no vaccine for HIV, but every year we get a new flu vaccine, so that we just know a lot more about it and how to produce those. So what are the special challenges in developing a vaccine for coronavirus?
So like you said, we know a lot about influenza. The vaccines are great, but they definitely need to be improved. And so there's 100 years of influenza research as a backdrop for understanding and dissecting immune responses to influenza. There's very little historical context for coronaviruses. The field went from tens of labs to thousands of labs overnight. And as I stated a few times, it's we stand on the shoulders of giants and of data that historically existed, and that data doesn't exist. And everything from developing animal models, there's a lot of controversy over how good a non-human primate animal model is, because they don't get very sick from coronavirus, right? So that's maybe not the best model to study this. We also don't understand why it infects some people and they become very sick, others don't, some people are asymptomatic. So there's just not the backdrop to know what the proper correlates are that we should be looking for, and then the demographics and how to vaccinate and treat them appropriately.
So the spike protein, of course, is the target for all the vaccine work. And in both HIV and flu, that's a glycoprotein. Is the spike protein in coronavirus a glycoprotein, and does that present special challenges for the vaccines and antibody work?
So absolutely, I totally glossed over that in order to keep this fairly high level, but the surface of the spike protein, just like HIV and influenza, is covered in glycans or sugars. In influenza and in HIV, it's well appreciated that these are mechanisms to prevent antibodies from binding, so they're evasion mechanisms. In coronavirus, it's actually highly debated whether those glycans play a role in immune evasion. And for the RBD antibodies, glycans don't seem to play, a real role. The ACE2 receptor binding site is not glycosylated, at least where ACE2 binds. So we don't know. I think the challenges that glycans present are in studying them. So, in our lab, we're developing new methods structurally. There's a lot of cutting edge mass spectrometry that's being done to study the chemical composition. And then from a manufacturing standpoint, you can't use E. coli or yeast to make these. You have to use typically mammalian cell lines. And so that adds cost and other complications to scaling up, especially for protein subunit vaccines. So they're a critical component of the vaccine, but there's sort of an unknown element of how they impact the immune responses.
So one of the things you mentioned is that you're looking at the complexes of antibodies, different kinds of antibodies, with the coprotein. So are you going to be able to use that information maybe to help guide the design of a second generation vaccine?
Absolutely. So that's the pipeline that we employ for all these other vaccine programs, where, again, from infection or experimental vaccines, we look at on-target and off-target antibody responses and the sort of ratio between them. And then what we go in and we sit in front of a computer and design mutations or other modifications of the protein, these spike proteins, that will turn the focus on these off-target responses off, and then also amplify the on-target responses. And so, again, that's what I was saying about lessons learned here will allow us to sort of understand where we should be focusing our efforts for future pandemics, future spike proteins. And that pipeline is very well established now. We just need to populate it with data from infection and experimental vaccines.
Another kind of theme question that a couple of people have asked in a slightly different way, but what about mutations in the virus that would affect the spike protein? What would that do, and is that a challenge for developing a vaccine?
Right. So evolution of viruses is a key concern, especially for RNA viruses. Influenza uses mutations all the time, seasonally, to escape the pre-existing immunity. Here, there's not enough data to know as far as the type of mutations that are being made. I showed one mutation that seemed to not so much arise from escape from an antibody response, but seems to make the virus more pathogenic or spread more easily. So those do create problems. However, and again, maybe an argument for not just going after the receptor binding domain is that if you have multiple antibodies that bind to multiple places in the spike, right, each one doing a different, having a different mechanism of inactivation, means that it would take multiple mutations for the virus to escape that. And really, canonically, that's how most vaccines work. You generate a diverse set of antibodies that target multiple functions in the virus, and it really renders its ability to mutate and escape useless. So I think that's something that will come out from the epi studies, will come out from, once we see if people become reinfected or reinfected with a different strain that emerges.
Another question a couple people have asked is about ACE2 inhibitors. So what's up with drugs that probably there's some people in the audience that are taking, and they're binding to the ACE2, and does that in any way affect, enhance, or inhibit the interaction of the spike protein with the cell entry?
So great question. That's really outside my area of expertise, but certainly anything that binds to the receptor is going to be competitive with the virus. But of course, the virus is decorated in those spikes. It has a corona of spikes around it. And you have a lot of ACE2 on the cell surface. So there's probably sort of a balance in terms of avidity and binding and competition. And so if it was just a monovalent, one spike binds one ACE2 and that's how the virus gets in, then that might be mechanistically, the small molecular drug might block that. But if you have a lot of spikes and a lot of ACE2, that drug is maybe not be saturating enough to do anything for the virus or prevent the virus from getting in.
Okay. All right. I think we're coming up on an hour. And there's probably another 30 or 40 questions that we could get to, but I think in the interest of time, we should just wrap it up. So thanks a lot, Andrew, for the seminar and for the questions. I do want to acknowledge our sponsor in this front row lecture series, which is KPBS. And I'd like to give a plug for the next one. So Andrew's lecture is the last in our series on the coronavirus. And we're going to transition to highlight some of the other basic research and translational research projects going on at Scripps. So on July 1st, we'll have a tag team effort from Hugh Rosen and Luke Lairson. And they're going to be talking about therapeutic approaches to combating multiple sclerosis. So look forward to that. We have front rows scheduled for the rest of the year. We'll have at least one per month. So come back to the Scripps website and check it out. If you'd like to watch this, maybe you had a little internet trouble, you can watch this in any of our previous lectures on the website. So go to front row at scripps.edu, and you should be able to find your way to the front row webpage. Thanks very much for staying on and engaging, and we're really happy to help disseminate what basic science can do for human health in this country. Thanks a lot, Andrew.
Thanks.
Bye all.